Good evening, class. So, thus far, our conversation about the sensory systems has been primarily on um, vision and audition. What I'd like to do is expand our discussion into the chemical senses, which consists of olfaction, your sense of smell, and gustation, your sense of taste. Um, and I'm sure you remember playing this game as a child, right? People would ask, it's slightly morbid, uh, you know, if you had to, which of your sense systems would you lose? And most people tend to choose something like olfaction, right? Because who wants to smell the horrible smells that there are in the world? Um, but our chemical senses are actually quite important in terms of our survival. And this combination of gustation and olfaction is what contributes to our ability to detect flavors in food, for instance. So the chemical senses were the first sensory systems to develop a chemical sensitivity that would enable uh, animals to find food and avoid certain kinds of danger and even to locate mates. And there's some discussion that the chemical senses are actually the foothold to emotions, which we are going to talk about in our next lecture. This moving towards of a less complex organism, this moving towards something that's positive and physically retreating from something that's negative or beneficial to survival and adaptation versus harmful for our adaptation is the foundation for complex emotional systems. But we'll get to that next discussion. Okay, so let's talk about chemical coding. Um, Coding in the sensory systems could follow two principles theoretically. One is the label line principle, and here each receptor responds to a limited range of stimuli and sends a direct line to the brain. So it's quite possible that vision and hearing work primarily through a label line principle, right? So you have a receptor type that's a cone that responds to a limited range of stimuli, so not the full electromagnetic spectrum, but this limited range, and sends that information to the brain through a direct line, right? We have those receptors to bipolar to ganglion cells. Chemical coding is most likely, the chemical senses are most likely coded um, through what's called a cross fiber patterns. So here each receptor responds to a wider range of stimuli and contributes to the perception of each of them. And it's thought that the timing or the pattern of the responses allows the cortex to disambiguate some meaning from that neural response. So the vertebrate system probably have no pure label lined codes. So if you think back to vision again, when we talked about those peak absorption spectras, that short wavelength cone type was still absorbing some wavelengths outside of its peak absorption rate, but was primarily responding to like short wavelengths, for instance. But there still needed to be a combination of responses in order for the brain to determine or to perceive a particular type a particular hue. So auditory perception, color perception, they rely on label line codes, but there's also cross fiber responses, right? The pattern of response and the combination between multiple cone types that influence what it is that we perceive. Taste and smell stimuli are going to activate several neurons. And so what this means is that the response of a single neuron depends on the context of responses by other neurons. So again, it's pattern and timing of the response within a particular neuron. That timing and response of a particular neuron is being influenced by neighboring neurons, and that's going to inform what one perceives. So let's start our discussion with taste or gustation. And taste has one simple adaptive function. It's to tell us whether to swallow something or to spit it out. And this is actually quite important if you think about this, that the taste receptors are really this line of defense before something enters into your body, right? Before you take something into, the deeper areas of your body. We need that information to know whether or not something is safe to consume or it needs to be spit out. And we like sweet tastes, even in infancy. Sweet tastes good to us because glucose is the brain's primary source of nutrition. Whereas, we dislike things that are bitter and sour, but we will ingest these items in small amounts. And our response to bitterness in particular, because, right, that's toxins that plants release. They're trying to make it so that other animals don't consume them. So they increase their level of toxicity. It creates this bitter flavor. The level of toxicity in kale, for instance, is not going to harm us. So we can accept that in small amounts, but I don't think there's a pregnant woman on earth who will have cravings for a kale salad when she's pregnant, for instance, right? So when females become pregnant, they tend not to crave and actually become repulsed by the bitter flavor, because again, that's a sign of toxicity, and we want to protect this developing fetus. And it's shown that people vary in their liking for salty flavors. Now let's just go back to this idea of whether to swallow something or spit it out. This last line of defense before you bring something into your body. I put on Moodle an article that some colleagues of mine and myself, we worked on this study back when I was an undergraduate, actually. And we were looking at the perceived intensity of a smell when a liquid is colored or not. And what we found, which was really interesting, is if somebody's asked to smell clear fluid and like with food coloring to make it much brighter color of red, and they're asked to smell it and rate the level of fruitiness that they're detecting, the color, that combination of the visual with some odor molecules present will actually make that smell more intense. They rate it as fruitier if it's got the food coloring in it than just the clear liquid, even though the concentration of odorants is the same. if we put those beverages in cups with lids and straws, and we ask participants to bring those fluids into their mouth and smell it retronasally or from inside the mouth, you don't get that effect. And what we posited in the discussion is that because when something's already in the mouth, it is more important that the perception be more accurate, more true to the physical makeup of that stimulus, that we actually lose the effect of color on odor intensity when smelling something once it's inside the mouth, which is pretty cool. So you can read that article if you're interested in what I was doing as an undergrad. It's pretty cool. It's fun. Anyway. Okay. So taste. So taste is the perception of the stimulation of taste buds. And these are receptors on the tongue. Like I was saying before, our perception of flavor is actually a combination of both taste and smell. Taste and smell axons converge in the endopiriform cortex, and again results in what we call flavor. But taste, we have primary taste: sweet, salty, bitter, sour, and what's called umami, which we'll get to in a minute. And we may have a sixth, which is just the smelling of fats. That sensory component, that's taste. And again, the flavor of something is this combination of taste and olfaction. So the receptors for taste are actually modified skin cells. Our bodies are quite, what is the word I'm looking for? Like economical. Once there's an adaptation and it works, it could be slightly adapted to serve other purposes instead of something completely new. Some sort of new generation. It's just that we adapt things that work for one purpose to work for other purposes. Taste receptors have excitable membranes that release neurotransmitters to excite neighboring neurons. So again, this is the cross fiber principle. These neurotransmitters, when excited, are going to stimulate neighboring neurons. And taste receptors are replaced every 10 to 14 days. We'll see that olfaction receptors are regenerated as well. Taste buds are located in papillae, which are structures on the surface of the tongue. Each one may contain up to 10 or more taste buds, and each taste bud contains approximately 50 receptors, which again, are communicating and sending messages to their neighbors. And most of your taste buds are located along the outside edge of the tongue in humans, on the tip, some are on the back of your tongue. So a really fun exercise is to take something like, salt water or something sweet, like honey, and, get it on a Q-tip, and you take the Q-tip and rub it along the middle of your tongue, and you're not going to have a taste sensation because there are no receptors there. However, if you begin by taking that Q-tip and rubbing it on the side and then moving it to the middle, it's kind of like the filling in of the blind spot, right? You'll be able to have a taste perception as it moves to the middle of your tongue where there's actually no taste buds there. And so have fun, play with a Q-tip soaked with some sort of a solution, so you can try this out. So there are procedures that can alter one receptor but not others. This has allowed us to identify the taste receptors, and like I said, um, there's five primarily, possibly one that's sensitive to fats. Um, Adaptation, which is a good term to know not only in this context, but in the context of other sensory systems. Adaptation refers to the reduced perception of a stimuli due to the fatigue of receptors. So if you have a receptor that's sensitive to sweet, chemical compound for sweet, okay, and you stimulate those receptors, so there's a sweet substance that's stimulating those receptors, eventually that'll fatigue. And so you'll need more of the stimuli or more intense stimuli to be able to even detect sweetness. However, if you're adapting sweet receptors, right, or you're fatiguing them, but then you put, a sour or bitter substance on those papillae or across those taste buds, there's no adaptation to that newly introduced chemical compound, right? So that would be, if that happened, if there was a reduced response to bitter from stimulating sweet, that would indicate that those aren't individual receptors. That would be a type of cross adaptation. So here are your primary tastes. We have sweet, sour, salty, and bitter. There's evidence that suggests that there's a fifth type of glutamate receptor called umami. It is the MSG. It stimulates glutamate receptors. And again, you might have a receptor that is sensitive to fats. These receptors are stimulated, or I should say activated, to send their communication in different ways. The receptors that are sensitive to salt, they open up sodium channels and allow ions to cross into the membrane, and that results in an action potential. Sourness receptors close potassium channels when acids bind to the receptors. It results in a depolarization of the membrane. So again, depolarization is it becomes less negative on the inside of the cell. And sweetness, bitterness, and umami receptors activate G proteins. So they release a second messenger in the cell when a molecule binds to a receptor. So the saltiness receptor is ionotropic, right? It's active like ionotropic receptors. Sweetness, bitterness, and umami are metabotropic functioning with the release of a second messenger or G protein. Different chemicals result in different temporal patterns, right? So different timing of neuronal activity in the brain. So taste is a function of the type of cell that's active, right? Which receptor type is it? And the rhythms of cell activity. And here are different types of receptors firing in different timing patterns. Again, that information allows the brain to perceive the sensation. Now, this this fact right here that bitterness receptors are sensitive to a wide range of chemicals with varying degrees of toxicity gives us an indication, again, of how important it is to be able to perceive bitter as a taste sensation. There's about 25 types of bitter receptors that exist. Again, bitter is an indication of toxicity. We want to be able to identify that as the last line of defense before we ingest anything. Other taste cells contain only a small number. Most taste cells contain only a small number of these receptors. And again, wide range of harmful substances, but not highly sensitive to a single one. Again, we want to make sure that if there's a chemical compound that is toxic, that we're able to have some messages about that going to the cortex. Different nerves can carry taste information to the brain from the anterior 2/3 of the tongue, then from the posterior tongue and throat. And these nerves project to a structure in the medulla known as the nucleus of the tractus ciliaris. And from there, projections are sent to various parts of the brain. Okay, so this is your pathway information. Now, your primary taste cortex is called the insula, and it's located right here. And very interestingly, when we are in an emotional state of disgust, when we perceive moral disgust, there is activation in the insula as well. So it's not just responsive to taste information, but it's also responsive to disgust as an emotion. Various areas of the brain are responsible for processing different taste information. The somatosensory cortex responds to the touch aspects of taste. The insula, again, is the primary taste cortex, and each hemisphere of the cortex is also responsive to the ipsilateral, same side, of the tongue. So this slide nicely, captures what we're talking about, modules and distributed representation, right? So there's a primary taste area of the brain. That's part of its function. We have this taste module, but there are other aspects of the items that we put into our mouths that have a taste that need to be processed by other areas of the brain. This slide, for instance, says the touch aspect is processed in the somatosensory cortex. Genetic factors and hormones can account for some differences in taste sensitivity. Like I was saying about, pregnancy and the sensitivity to bitterness, how that increases. Some people are super tasters. They have a higher sensitivity to all tastes in the mouth and mouth sensations generally. And it's related to the number of papillae near the tip of the tongue. So again, super tasters have higher concentration of taste buds at the tip of their tongue, whereas this is the concentration of taste buds or papillae for non-tasters. And there's a genetic component to this. Okay, so that's taste. Now for the other piece of this flavor dynamic is olfaction. and it is our sense of smell, and refers to the detection and recognition of chemicals that make contact with the membrane that's inside of our nose. And olfaction is a critical sense for most mammals, for finding food and finding mates and for avoiding danger. And this is another one of the sensitivities that's going to be informed by hormones and genetics. We can actually follow scent trails to some extent. Here's a picture of a participant in a lab study where they're able to, with the rest of their senses unavailable to them, actually follow a scent trail. It is not the most sensitive system that we as human beings have as opposed to other animals, but it is a important sense for our survival. Okay. So like I was saying before, this sensitivity to smell and its relationship to hormones, humans tend to prefer the smell of potential romantic partners who smell different from themselves and their family members. And whether or not somebody is taking oral contraceptives or is on birth control, that could impact the sensitivity or the preference of smell of a partner. Women smell different to males when they are on when they're in various stages of their menstrual cycle. It's wild. I should post a video. I forgot to put it in the slides, but they have these instead of like speed dating, what some groups started to do is you have people blindfolded and you're actually smelling each other's armpits, and you're giving a rating of attractiveness based on the smell of the person's armpits. And then you also fill out a survey for all of the kinds of traits that you're looking for in a partner. Yes, but you're rating attractiveness with this blindfold. And they've, taken sweaty shirts from women who are in different stages of their cycle, and the women who are closer to ovulation smell more attractive to heterosexual men. Interestingly enough, you take the sweat armpit secretions of a man and you have a female smell it, and it actually creates a stress response. So it's actually a sign to us of danger, whereas for males, it is an indication of attraction. And we'll talk about this more when we talk about evolution. It's really interesting how our mate preferences and our ratings of attractiveness and all these things shift based on where we are in hormonal cycles, right? Males have hormonal cycles of testosterone highs and lows as well. And even these indicators of attractiveness are going to shift depending on, like, were we looking for long-term mates? Are we looking for short-term mates? It's very interesting data, and it seems to a lot of these patterns seem to hold for homosexual individuals as well. But we'll talk about that when we get to that chapter. It's pretty interesting research. So again, the idea is, like, when you're snuggled up to your mate and they're smelling great, what you are smelling is their immune system. They are less likely to be related to you, and it is more likely that your offspring will be immune to numerous diseases. So your olfactory cells, They line the olfactory epithelium in the rear of the nasal passage and are the neurons responsible for smell. The receptors are located on cilia, which extend from the cell body into the mucous surface of the nasal passage. And vertebrates have hundreds of olfactory receptors, which are highly responsive to some related chemicals and unresponsive to others. So it sounds very cross fiber principle to me, sensitive to a much wider range of stimuli. So here in this figure, you can see the receptors and where the olfactory bulb is located, as well as the olfactory nerve. Remember that olfactory information does not go to the thalamus before it goes to its primary processing area. The proteins in the olfactory receptors respond to chemicals outside of the cells and trigger changes in G proteins. So it's a metabotropic response. And these G proteins then trigger chemicals that lead to action potentials. So from the olfactory receptors, the information is carried to the olfactory bulb. Chemicals that smell similar are going to excite neighboring areas, and chemicals that smell different excite more separated areas. So think of that similarity with the visual system when we talked about the columns of the cells, and I was talking to you about how our orientation cells, right? If, you know, the cells that respond to these vertical bars of light, right? If you change the angle of the light that a cell responds to, it's going to live right next to one another. And cells that process the same type of bar of light are going to be in the same column. So coding in the brain is determined by which part of the olfactory bulb is excited. Again, parallels to the other sensory systems. The information goes from the olfactory bulb to the cerebral cortex, where messages can be coded by location. Olfactory receptors are replaced approximately every month. They are subject to permanent impairment if there's a massive affront to that sensory system. So it's possible to do damage that they won't regenerate. Individuals who lack a sense of smell, it's called anosmia. And again, individual differences in olfaction exist. Generally speaking, women detect odors more readily than men, and their brain responses are stronger. However, menopausal women's sense of smell starts to match that of males, and also females prior to puberty. Their detection and brain response is similar to males. Right, so it's this ability to detect odors become more sensitive for women who are in the window of reproductivity. So it seems to be influenced by hormones. Now, pheromones, very interesting, right? So one of these questions is when we smell the armpit of our beau or our, this person we're attracted to, and we're smelling their immune system, how is this different or similar to pheromones? Well, what pheromones are is they're chemicals released by an animal to affect the behavior of others of the same species. And, if you've ever seen a cat in heat, There's a particular time where these animals are receptive to mates, and these pheromones are released and they attract the mates of the opposite sex. And the stimulation or the reception or detection of the pheromones is happening in the VNO. And there's receptors located, olfactory receptors, and they're responsive to pheromones. Well, what's very interesting is that in humans, the VNO is quite small and it has no receptors. So we may be responding to some pheromones through receptors in the olfactory mucosa as opposed to the VNO. Another example of odorants of these chemicals having an effect on other members of the species is the synchronization of menstrual cycles in women. They did a study where they had a group of women smelling the secretions of a particular woman, and they all synced up their menstrual cycles to the donor of the odorant, to her cycles. So lastly, just want to, we've heard this concept synesthesia before. It's the experience of one's sense in response to stimulation of different senses. It's estimated 10% of 6 and 7-year-olds, so it develops gradually. There's a genetic component to synesthesia. It appears more in artists, and it's one of those, what comes first, the chicken and the egg? Does having synesthesia have somebody more prone to be creative and explore their world and represent their world artistically? Or what is this common factor involved with the arts and having synesthesia? We listened to Ramachandran early on in the semester talk a little bit about synesthesia, and he mentions that it's most likely similar to metaphor, where this cross-representation of sensory information helps us get a fuller meaning or descriptive picture of our environment. So we're all synesthetes in some kind of way that we can make these associations between variables or things out in the environment. But true synesthetes have a perceptual experience. When they see a number, it's colored. All 4s are yellow, all 7s are green, and we know that this is actually their experience because, I don't know if you've seen these things in one of your other classes or maybe on Facebook. They'll put up, a whole bunch of, let's say, emoticons, and there's like one face that's slightly different, and you have to time how long it takes you to find it. Or there is a bunch of numbers, and only one number is slightly different, but the form is pretty similar. Synesthetes can find it very quickly because that number is a different color and it pops out, right? Color is an attention grabbing feature and it'll pop out of the stimulus array. So people who are synesthetes, who have color number synesthesia, can find these numbers very easily. So we are more confident that it's not just a farce, that they're making this up. They're actually having a perceptual experience that's a mingling of sensory information. And it's quite possible that it is a genetic mutation that affects the pruning. So there's not as distinct modules for the sensory information. There's more neuronal connections among them, which leads to this phenomenon. Yeah, so that is the rest of the sense systems. Well, I hope you enjoy that. I'll see you on Wednesday. Bye.